We are Group B05, and we examined the case study Fighting Malaria with Engineered Symbiotic Bacteria from Vector Mosquitoes. In an effort to combat malaria, scientists have begun experimenting with genetic modification of mosquitoes. However, this approach to genetically modify the insect's genome has proven to be difficult and likely impossible for several reasons. One in particular is ensuring the modified mosquitoes outcompete their wild peers. Instead, experimenters began to focus on what resides within the gut of mosquitoes: Plasmodium and bacteria. A malarial mosquito is a flying factory for Plasmodium, a parasite that fills its guts and storms the blood of every person it bites. Additionally, bacteria reside in the mosquito gut, and they are being loaded with genes that destroy parasites. The fact that bacteria also live in the mosquito gut is extremely advantageous, for the most vulnerable stages of Plasmodium development occur in the lumen of the mosquito midgut. The following presentation describes an experiment by Wang in which engineered bacteria strains were used to inhibit the development of the human malaria parasite in mosquitoes.
The results from this experiment showed that 5 potent anti-Plasmodium effector molecules secreted by P. agglomerans were able to efficiently inhibit P. falciparum development during early sporogeny. P. agglomerans were engineered to make several proteins that turn the mosquito's gut into a hostile zone for Plasmodium. Wang and his team found that the engineered bacteria slashed the number of oocysts in the mosquitoes by 85 to 98%. Just 14 to 18% of the insects carrying the defense microbes became infected with Plasmodium after drinking contaminated blood. Recombinant P. agglomeran strains do not affect mosquito lifespan. There were no negative impacts on the mosquito fitness. P. agglomerans were reported as a dominant symbiotic bacterium in various mosquitoes used to fight off malaria. P. agglomeran strains have been used as a microbial control agent against the plant disease fire blight in the U.S. The effector molecules from this study are equally effective with A. gambiae, African mosquitoes, and A. stephi, Asian mosquitoes. The midgut of these mosquitoes serve as a prime target for blocking parasite transmission. P. agglomerans was engineered to make several proteins turn the gut into a hostile zone for Plasmodium. The bacteria could be transferred to a village via baiting stations. Baiting stations are potential loading docks of the genetically modified bacteria to the mosquito. In here contain cotton pads, which are soaked with the bacteria suspended in a 5% sucrose solution. The mosquitoes will then fly over, sip the solution, then become loaded with the anti-Plasmodium defense. It's simple and fairly cheap, but since modified bacteria would be using energy to produce anti-Plasmodium molecules, mosquitoes would have to be exposed to the large numbers of these genetically modified bacteria in the field for the bacteria to stand any chance of becoming a major portion of the microbes that reside in the mosquito gut. Some are afraid that genetically modified bacteria cannot be contained, and thus with time end up in our food chain and onto our food plate. Some of these opponents are leaning along the lines of naturally occurring bacteria.
Let's briefly go through the advantage of this new technology. First, it is not using drugs to kill mosquitoes, so there will be no more drugs persistence and pollutions. It is practical not just in lab, but also in the wild, which means it can be widely applied in the world. Second, the midgut of mosquito is the bottleneck of the development of Plasmodium, so it is the most efficient to kill them in the midgut. Third, there will be much more engineered bacteria produced after a blood meal, so they can secrete a lot of special protein which can kill Plasmodium. Also, these bacteria will not affect the longevity of mosquitoes, which means they are benign. But this new technology still faces some challenges. First, because the engineered bacteria will have to spend valuable energy on producing the anti-Plasmodium molecules, they might be less competitive than normal bacteria. Second, many people are deeply opposed to the use of genetic modification, and such antagonism is especially high in many African countries where malaria is endemic. Third, while such bacteria may not pose a hazard to humans and animals, the work required to be proved. So there are still a lot of work need to be done before this technology is mature.
And finally, a take-home message. We have discovered a foundation to combat malaria through the use of genetically modified symbiotic bacteria, but still face a challenge through population approval and regulatory issues.